All right, here we go. This is the screencastify for class on February, let's see, 17th. And so right now we're getting ready to do some notes. And so pause this until we get going on something else.
All right. I have a few more pounds than I should, but still eat like I'm 17, so catches up with you just so enjoy it right now. You can eat as much as you want, right? All day, every day. So yeah. All right. So. Okay.
Sorry about that. I had just thought I recorded everything, so that last 20 minutes. Sorry, we just went over notes. I apologize. I was a dope, didn't hit the right button.
We did temperature. And there's copy right here. It's kind of crappy,
all right,
but it'll
Mr.
work.
Wolf is getting ready to go over.
Is it recording this?
So if you do not have your own printed
yes, now
version
it's
of
recording.
This,
thank you.
So that you can write the answers down.
And so he's going to go over that worksheet that we're supposed to have done for this week.
Once and twice. And again, take that out if if you have your own, take it out so you can check your answers.
I think my laptop is broken.
All right, here we go.
I think yours.
The answers.
The battery. That's true.
Check your answers, write correct answers down. Samuel, are you ready?
This is the paper I'm talking about. He's about ready to go over the answers.
I said I'll go through these pretty quickly with you here, maybe. Why is it not moving? So one is D, the sequence. It goes glycolysis, Krebs cycle, electron transport chain. The second question is B. What happens to the carbon atoms that originally were in the glucose? They eventually end up as part of carbon dioxide and then end up in the atmosphere. So if you track those, that's where they would eventually end up. Requiring oxygen is aerobic. Total ATP produced by one molecule of glucose during aerobic cell respiration is 36. Sometimes you'll see it's 38. It kind of just depends on whether they're including the two from glycolysis or not. But the maximum you could get is 38, but sometimes you see it's 36. It usually doesn't produce that many, but that would be the maximum you could get. The next one is all of those. So what are the following produced during the Krebs cycle? It produces all those things. There's some ATP produced, there's some NADH, FADH2, and CO2. Write on your paper, make this note to yourself. These are the two in the middle here. They are energy carriers. So they take the energy from the Krebs cycle, pass it along to the electron transport chain. And this energy originally was in the glucose molecule. It works its way through these processes and gets trapped in these molecules. Then they get sent on to the electron transport chain. So those two things, NADH and FADH2, are energy carriers. Oxygen, final electron acceptor. Hydrogen ions are what moves across the membrane of the mitochondria. That causes, it's actually diffusion. If you remember from high to low, things diffuse. So the hydrogens diffuse across the membrane, actually provide the energy to make the ATP. And then the next column up, electron transport, just makes the most ATP. A, that's what happens during the electron transport chain. Energy from the moving electrons causes hydrogen to go into the inside of the membrane of the mitochondria. Don't really worry about the stuff in that box there. That's things we didn't really get into. Same with this one. It is citric acid, but we didn't, you don't need to know that. This is important on the back here. Where does this stuff happen? So that's a mitochondria. Place where glycolysis happens would be E, which is outside the mitochondria. It doesn't happen in the mitochondria. Glycolysis happens in the cytoplasm. C, which is right there in that membrane, it's a place where enzymes for electron transport chain are at. And B, between the two membranes, is where the hydrogen ions move. They move into that space. So, all right. And then all this stuff here, I'll give you a second to write this down. So again, this top is the name of the process. Glycolysis happens in cytoplasm, splits glucose. It's anaerobic, doesn't require oxygen, makes two ATPs. That's the first step, called glycolysis. And then after that, you get to the Krebs cycle, which occurs in the mitochondria. It's aerobic, makes CO2, makes these energy carriers called NADH and FADH2. Okay, this is called the Krebs cycle. Again, you don't have to know all these little steps and all stuff, just the stuff that's written in pink or red there. And lastly, the electron transport chain. And it occurs in the inner mitochondria, uses oxygen, makes ATP and water. Crap load of ATP, 34 of them. So there you go. All right. So at this point, you're going to do another experiment where you're going to study your own production of actually the Krebs cycle. You're going to be seeing the evidence of Krebs cycle happening in your own bodies. So I will pass this on. Mr. Urbanek will show you and tell you what you're doing with this lab.
Okay, you're going to work in groups of 2. More than 2, don't do it. And what you're going to need to do is come up here and get some items. So make sure you're paying attention. You need a 100 milliliter graduated cylinder. You need a 10 milliliter graduated cylinder. You need a flask. You need the directions. That's right there. You need a straw. And you need this clear fluid, which is sodium hydroxide. Sodium hydroxide, NaOH. And you need bromothymol blue. Now, there might not be enough bottles of the bromothymol blue to go around, or the NaOH to go around. You can share. 4 people are working at a table, 1 in the middle. That's really all you need for that. And so everything else, make sure you grab. This lab is not hard. If you can read and you can breathe, you can do this lab.
Literally, that's what you need to do.
The problem is, my students will not follow the directions. So where it says fill a 600 milliliter beaker to 350 milliliters of water, use the graduated cylinder. They don't use the graduated cylinder, they just go by what's written on here. That's the mistake. Don't do that. When you measure things properly, you get better results. That's why I'm telling you to measure these things. As you can see, I did this, and that's around 375. These are very general types of measurements. This is accurate, and that's the problem. If you want to be accurate, you want to get better results, follow the directions. And basically, you're going to make a solution like this. And you're going to do what it says in the directions. You're going to blow into it, and the carbon dioxide is going to come out, and it's going to change its colors. And that's what this is. It's different color. And so then you'll have to add sodium hydroxide to bring it back, counting the drops, back to this color. Now notice, when you come over here, I have my example on white paper because it looks different on a black background versus a white background. This has got a greenish color, and when I'm holding it, I can sort of see green to it, but when I put it on the black table, it's black. So again, put it on white. It makes it a lot easier. You can definitely tell that that one's more blue. So again, if you can breathe and you can count and you can measure, you can do this. And so just read through it. It's really not hard. A little hint though, when you're blowing into it, just like when you were a kid, you're blowing bubbles into the milk. You know how it bubbles up? If you put your hand over it and you put the straw through your hand and you're blowing, you don't splash all over your face. So it might not want to be a bad idea just to put your hand like that as you're doing it. And you just breathe normal, you're not trying to go crazy and blow the bubbles absolutely everywhere. Just normal breathing, not a big deal. Then you exercise. I think it says for 3 to 5 minutes. It does. 3 to 5 minutes. That means up and down the stairs for 3 to 5 minutes. Yes.
Get after it, people.
Get that heart rate up. Remember the formula. You got to use that oxygen to get stuff going. If your heart rate is not moving, you haven't gone enough.
I'm dead.
So you have to make sure you're breathing, and then you breathe into it and you change it and see what kind of differences there are. Now I'm leaving mine up here. You'll need to compare your solution to this one after you've done the breathing, because you have to bring it back to normal. So you have to count the drops, bringing it back to normal. This is the normal one. Compare yours. Bring yours up. Say, oh, mine's too light, mine's too dark, or whatever. Just make your adjustments. So that's what you need to do. It's not a hard lab to do. Just read and follow the directions. Groups of how many? All right, come on over and start grabbing stuff. Yes, you can go next door over here. That's a good idea, actually. I'll even turn the lights on.
Whoa.
You can do it.
Measure accurately. Good move. Good start. Hello.
Oh, boy. Good one.
Sound good.
Who dropped it?
All right, we're doing a lab. Pausing.
Record that real quick.
What stuff?
It's on.
Whatever
There
are
we
you
go.
talking
Thanks,
about?
dog. Thank you.
Is there just a lot of people missing today? About 5.
COVID.
Let's put in that base tray, yeah.
Shut this down.
Well, should we go through that chart again?
What?
All right, these are the notes that I sort of didn't get copied before. So it's at the end of the period where I said before I screwed up. Well, here are all the notes again.
Okay, so first thing we talked about was ATP. So just understanding what that is and what its job is in cell respiration. So ATP, it's basically like it says here, it's the key molecule that actually carries the energy in our cells, and so it literally is what provides the heat to make things happen in the cell. So a lot of times we talk about glucose being the source of energy, which is true, but the glucose actually gets converted into ATP, and then the ATP provides the energy. So, yeah, I thought this would happen. Hold on.
ATP stands for adenosine triphosphate. Make sure you know that.
It's working. I just didn't.
Crazy machine.
Yeah, why is it doing that? All right.
Yeah, why is this thing up here?
I have no idea.
Oh, that was easy.
All right. So, like Mr. Urbanek just said, this ATP stands for adenosine triphosphate, which is that name right there. And
Before you go any further, think of ATP like a little rechargeable battery. When you have these 3 phosphates here, it's all stored up, but when you need it, one breaks off, a little energy, a little heat comes off. Then you have ADP, then you eat, energy gets put back into it, then it's ATP. It's just constantly like a little rechargeable battery.
perfect. So there you go. So yeah, adenosine triphosphate. The ADP, like Mr. Urbanek just said, is adenosine diphosphate, and it just cycles back and forth. So again, this is the molecule that provides the direct energy to the cells, and it, like he said, we're going to talk about the recharging of this battery. All right. Switch over here.
The thing to remember is that this is happening in the cell in the mitochondria. That's where the mitochondria is going to be using the different chemicals and things to make that ATP for us.
This is what this is talking about right here. So again, cell respiration, making ATP, breaking down the glucose that we eat. And then there's two types or two stages. One is anaerobic, which is without oxygen, and that happens in the cytoplasm of the cell. And then there's aerobic part of it, which uses oxygen and happens in the mitochondria. So those are the two things we'll talk about, is anaerobic and aerobic.
And you can see the mitochondria is all folded. So that means there's lots of places for all of these reactions to happen.
Okay. So this is a flow chart. Again, if you don't have this somewhere, you could print it. It's on one of those links. But if you don't want to print it or can't print it, copy this down. It's really, this is everything that's going on that we're wanting you guys to know. So well, first it always starts with glucose. In the beginning, you're eating stuff and taking in the energy you need to recharge your ATPs. This is called, the first step's called glycolysis. And that's this step of taking glucose and breaking it down and making this stuff called pyruvate and some other stuff. Again, we're not asking you to draw these, know what they are or anything, but just to know that this thing called glycolysis takes glucose and turns it into this stuff called pyruvate. And it also makes that rechargeable battery stuff. It makes two of those ATPs. And this is anaerobic. And it happens in the cytoplasm.
Remember, anaerobic, without oxygen.
All right. So then after that, there's sort of a fork. It can go two directions, or the process can happen in two different ways. So again, if you continue without oxygen or
With oxygen over here.
okay, so those are the two possibilities. So again, this is called anaerobic. Why can't I spell? Okay. Again, this would be either if it's a yeast or a bacteria that can't use oxygen, or if it's an animal that's not getting enough oxygen, this side of the process will happen. So let's go with green here. So this, all right, that's what we call fermentation. And that's the process of taking this pyruvate stuff and converting it to either ethyl alcohol, if you're a bacteria or a yeast, and some carbon dioxide. Or if it's an animal, and we talked about this in class, when you get that burning in your legs from working out too hard, you make this stuff called lactic acid because you're not getting enough oxygen to do this part. So your cells do this instead of dying from not having that oxygen. This makes 0 ATP. There's no ATP produced by this. And so if you're doing this, the only ATP you're getting are these two. So again, bacteria, yeast, or a person not getting enough oxygen, or a dog not getting enough oxygen, or a cat, or whatever, fish. Okay? Anything else?
That's good.
All right, so that's again called fermentation. I guess the one other thing we talked about too is just so you're aware, obviously ethyl alcohol is what is used in beer, wine, stuff like that. And so yeast as you probably know, yeast are used to produce alcohol for wine, etc. And what those things are doing is this glycolysis, and then this fermentation making alcohol and carbon dioxide, which gives the fizziness to some of that stuff too.
Makes the bread rise.
Right, or if you're using yeast for bread. But again, in either case, bread or wine or beer, you have to supply them with glucose, which is the bread, or in the case of wine, it's
Grape juice.
Or
when I make my wine, I have grape juice, and then I add my yeast to it, and then it breaks it all down into the alcohol and the carbon dioxide.
And then the big part of this, if we're talking again, an animal or a plant or a fungus that can use oxygen, or a protist, anything except a yeast or bacteria or an animal not getting enough oxygen, it's what's called aerobic. So after glycolysis, it goes into the mitochondria where these things occur. And the first step, this part, is called the Krebs cycle. And that's making carbon dioxide, makes 2 ATP, and it makes a bunch of stuff called NADH, which this is a big energy carrier. So the energy is kind of going from glucose to pyruvate to this. So there's a ton of energy stored here. But after the Krebs cycle, you have some more ATP. And then the last step is called the electron transport chain.
So this is going on all the time when you're breathing, because, we're breathing in the oxygen that goes into our cells, into the mitochondria. And of course, it's going to break down glucose and, do all that kind of stuff inside our cells. But yet, what are we going to be releasing? Carbon dioxide. We all know that. You breathe out carbon dioxide.
All right. And we might ask you, quiz test, coming up is, it's not a question like, when is the carbon dioxide? When are you producing carbon dioxide? And you would say during the Krebs cycle. So that's after glycolysis. So in the last step, the electron transport chain, that's when this NADH, all that energy is converted to 34 ATP molecules. So that is a ton of energy produced in this last step called the electron transport chain. And this is when your oxygen is used. So when you're breathing oxygen all day, every day, your whole life, this is why you're breathing it for this very last step, which is going to combine oxygen with some hydrogen and make water. And you breathe this out too. So, you're breathing out your carbon dioxide from the Krebs cycle and breathing out water from the electron transport chain. And that water is made from oxygen that you're breathing and some hydrogen that's left over actually from this NADH.
There's
stuff.
the hydrogen right there. Breaks off.
And that makes
Some
the water. And again, this is the important thing. The 34 ATPs, that's what's keeping us going and alive and doing everything we need to do. So again, if you can use oxygen, you're getting 34 plus 2 plus 2. It's a total of 38 possible ATP molecules. If you can't use oxygen, you're only getting 2 ATPs from this first step called glycolysis. I think that was pretty much what we went over.
And the formula.
Oh, yes.
And then
Forget that.
The formula for cellular respiration is coming up.
Maybe. I think. Yeah.
There it is. And
So we want you to know this.
of course, where does it happen? In the mitochondria. And what's it making? Of course, you're going to be releasing carbon dioxide. You're going to be releasing water. But what are you making? The big thing is ATP, adenosine triphosphate. ATP.
Again, you can talk about things like, the oxygen here is not used till the very last step.
Electron transport.
This is made during the Krebs cycle. This is made at the end with the electron transport chain too. So it's not like everything happens all at once, but we
Sorry about that.
use that equation.
And where does that come from? The food that you eat, the drinks that you drink, the little packet of sugar that you just chug down. And there it is. Hopefully you're going to watch this. Peace. And if you're watching this thing, you have a spinach lab that you need to do either Thursday, Friday, or Monday. Make sure you get it done. Peace out.